Hello, everyone, and welcome to Receptors Part 4, Enzyme-Linked Receptors. This topic is divided into 5 modules, so we can learn each concept step by step. In this first module, we will build the foundation by understanding what enzyme-linked receptors are, their general characteristics, how they are structured, and how ligand binding activates these receptors to initiate intracellular signaling. So, let's begin with the definition. Enzyme-linked receptors, also known as catalytic receptors, are a class of cell surface or transmembrane receptors. As their name suggests, these receptors are directly linked to an enzyme, or become enzymatically active when they are stimulated. They are activated when an extracellular ligand binds to the receptor. These ligands are usually growth factors, cytokines, or peptide hormones that carry important signals from outside the cell. One unique feature of enzyme-linked receptors is what happens after ligand binding. In many cases, the receptor itself functions as an enzyme, most commonly as a protein kinase. In other cases, the receptor does not possess intrinsic enzymatic activity, but instead associates with, or recruits, a cytoplasmic enzyme, which then initiates intracellular signaling. Regardless of the mechanism, the final outcome is the same. These receptors convert an extracellular chemical signal into an intracellular biochemical signal through enzymatic catalysis, most commonly by phosphorylation of specific target proteins. This is an important point to remember. Unlike G-protein-coupled receptors, enzyme-linked receptors do not rely on G proteins to transmit their signals. Similarly, unlike ligand-gated ion channels, they do not produce signaling through direct ion movement across the membrane. Instead, their signaling depends on enzyme-mediated reactions that activate a series of intracellular signaling pathways. Because multiple signaling proteins become activated one after another, the cellular response develops more gradually. As a result, enzyme-linked receptors generally produce slower but much longer-lasting effects, compared with ligand-gated ion channels. These sustained signaling pathways regulate several fundamental cellular functions, including cell growth, cell differentiation, cell proliferation, cell survival, apoptosis, or programmed cell death, and cellular metabolism. Because of these critical roles, enzyme-linked receptors are essential for normal development, tissue repair, immune regulation, and maintaining overall cellular homeostasis. Next, we will focus on one of the most fundamental aspects of enzyme-linked receptors, their general structure. Although enzyme-linked receptors belong to several different receptor families, most of them share a basic structural arrangement. They are single-pass transmembrane receptors, meaning that the receptor crosses the plasma membrane only once. Structurally, they can be divided into 3 major regions: the extracellular domain, the transmembrane domain, and the intracellular domain. Let us look at each of these components. The first part is the extracellular domain. As the name suggests, this portion of the receptor is located outside the cell, on the external surface of the plasma membrane. Its major function is to recognize and bind a specific signaling molecule, known as a ligand. The extracellular domain provides the receptor with its ligand specificity. This means that a particular receptor can recognize and respond to particular signaling molecules. Many enzyme-linked receptors are activated by relatively large signaling molecules, such as peptide hormones, growth factors, and cytokines. Examples include insulin, which binds to the insulin receptor, epidermal growth factor, or EGF, which binds to the epidermal growth factor receptor, vascular endothelial growth factor, or VEGF, which binds to VEGF receptors, platelet-derived growth factor, or PDGF, which binds to PDGF receptors. Other examples include erythropoietin and transforming growth factor beta, or TGF-beta. Therefore, the extracellular domain is primarily responsible for ligand recognition and binding. However, its role is not limited to simply binding the ligand. Ligand binding can also produce structural changes in the receptor and, in many receptor families, promote or stabilize the association of receptor molecules with one another. This brings us to the second important structural component. The second component is the transmembrane domain. This region connects the extracellular portion of the receptor with its intracellular portion. Unlike GPCRs, which contain 7 transmembrane helices, enzyme-linked receptors generally contain a single transmembrane A helix. This single hydrophobic segment passes through the lipid bilayer of the plasma membrane. The transmembrane domain therefore acts as an important structural link between the outside and inside of the cell. It performs several important functions. First, it anchors the receptor within the plasma membrane. Second, it maintains the correct orientation of the receptor, with the ligand-binding region facing the extracellular environment and the catalytic region facing the cytoplasm. Third, it can help transmit ligand-induced conformational changes from the extracellular domain to the intracellular domain. In some receptor families, the transmembrane region also contributes to receptor dimerization or other structural rearrangements that are important for receptor activation. Therefore, although the transmembrane domain is relatively small compared with the extracellular and intracellular regions, it plays an essential role in connecting ligand binding to intracellular receptor activation. The third major component is the intracellular domain. This portion of the receptor extends into the cytoplasm and is responsible for initiating intracellular biochemical signaling. This is an important feature that gives enzyme-linked receptors their name. However, it is important to understand that not all enzyme-linked receptors possess their own enzymatic activity. There are 2 general possibilities. In the first, the receptor possesses intrinsic enzymatic activity. In the second, the receptor does not have its own catalytic activity but is associated with an intracellular enzyme. Let us first consider receptors with intrinsic enzymatic activity. Some enzyme-linked receptors contain their own intracellular catalytic domain, which may include a kinase domain. The kinase or catalytic domain is activated following ligand binding and initiates downstream intracellular signaling pathways. Tyrosine kinase activity, as seen in receptor tyrosine kinases, serine, threonine kinase activity, as seen in receptors for TGF-beta and related ligands, guanylyl cyclase activity, as seen in receptor guanylyl cyclases, and tyrosine phosphatase activity, as seen in receptor tyrosine phosphatases. These enzymatic activities can modify intracellular proteins or generate signaling molecules, thereby initiating downstream signaling and ultimately producing a specific cellular response. The second possibility is that the receptor does not possess intrinsic enzymatic activity. Instead, the intracellular domain is associated with a separate cytoplasmic enzyme. A major example is the cytokine receptor family. Cytokine receptors do not have intrinsic kinase activity. Instead, they are associated with enzymes called Janus kinases. When the appropriate cytokine binds to the receptor-associated JAK enzymes become activated and initiate intracellular signaling. Therefore, enzyme-linked receptors can be broadly understood as receptors that either possess intrinsic enzymatic activity or activate enzyme-associated signaling proteins. Another important structural feature of many enzyme-linked receptors is dimerization. Dimerization means that 2 receptor molecules associate with each other to form a functional receptor complex. In some receptors, the receptors are mainly present as individual molecules, or monomers, in the resting state. When a ligand binds, it may promote or stabilize the association of 2 receptor molecules. This brings their intracellular domains closer together. The close proximity of these intracellular domains is important because it allows the catalytic regions to become activated. However, it is important to remember that not every enzyme-linked receptor is activated by exactly the same dimerization mechanism. Some receptors undergo ligand-induced dimerization. Others may already exist as preformed dimers and undergo a ligand-induced conformational rearrangement that activates the receptor. Therefore, dimerization is a common and important mechanism, but the exact mechanism varies between receptor families. Once receptor molecules are brought together and activated, many receptor tyrosine kinases undergo a process known as autophosphorylation. Phosphorylation means the addition of a phosphate group to a protein. In many receptor tyrosine kinases, each receptor molecule phosphorylates specific tyrosine residues on the intracellular domain of the other receptor molecule. This process is often referred to as trans-autophosphorylation or cross-phosphorylation. Why is this important? Phosphorylation can increase the activity of the receptor and, importantly, create specific binding or docking sites for other intracellular signaling proteins. These signaling proteins can then interact with the activated receptor and transmit the signal further into the cell. Examples of proteins that can interact with activated receptor signaling complexes include growth factor receptor-bound protein 2, SH2, domain-containing transforming protein, insulin receptor substrates, phospholipase C-gamma, phosphoinositide 3 kinase, and SRC family kinases. These proteins help connect the activated receptor to downstream signaling pathways. With this, we have completed our discussion on the general structure and activation of enzyme-linked receptors. We have seen how these receptors are organized into an extracellular ligand-binding domain, a single transmembrane domain, and an intracellular catalytic or enzyme-associated domain. We also discussed how ligand-binding, receptor dimerization, and autophosphorylation activate these receptors and prepare them to transmit signals into the cell. In the next module, we will move beyond receptor structure and explore how these activated receptors transmit their signals inside the cell, including the major intracellular signaling pathways and their specific cellular effects. So, stay connected for the next part of receptors. Thank you for watching, and I'll see you in the next module.